Okay,
All right, and I
so
made it.
I'm Carol Bornstein, and I'm one of the members of the board of the Southern California Horticultural Society, and welcome to this evening's presentation. I just wanted to share a little bit of information about the Society before introducing our speaker. We are a very friendly group of people who are plant lovers, first and foremost, as well as garden lovers. And we have these meetings once a month, usually live, but obviously not right now due to COVID. And we have a plant forum, as John mentioned. We also have other occasional programs for our members scattered throughout the year, field trips of one sort or another, visiting gardens and other places of interest. And if you became a member after listening into some of these Zoom meetings and wanted to join us, we would love to have your support because memberships help to enable us to offer these programs with wonderful speakers every month, as well as the other events that I briefly mentioned. And that also helps us to fund several internships every year to students who are pursuing careers in horticulture here in the Los Angeles area. So we'd love to have you join us. So now I'm going to go ahead and introduce tonight's speaker, Ernesto Sandoval. Ernesto has been working at the UC Davis. I don't know if the conservatory is actually part of the Arboretum or not. He'll tell us that. He's shaking his head no. Okay, so it's independent. Well, he's been working at UC Davis for something on the order of 30 years, various different positions, and now he is the director of the Botanical Conservatory, where he's responsible for growing thousands of different kinds of plants. So his plant knowledge, I'm sure, is incredibly vast and deep. And tonight he's going to share with us some of the interesting, fascinating stories about plants, why they do what they do, how they do what they do, from the inside out. So please welcome Ernesto.
Thank you, Carol. Thank you. So I'm free to go.
You are.
All right, great. So I'll share my screen here and see if this works out. And y'all can see that right there. This is my PowerPoint.
Yes.
Yes, that's visible. Great.
Yes.
Okay. Let me just move something out of the way so I can see better. Okay, yeah, great. So, previous, go back. Okay, so yeah, I'm here to talk about plant hormones, how they work, and for propagation and growth, and how they can help us grow our plants better by understanding them. And so I've done this talk many times. I did it on chalkboard a while ago, and I finally digitized it a number of years ago. And so I could give this talk now. I can even give it from a distance. So if y'all know of any garden clubs that would appreciate it, I'd be happy to share it along with some other ones. But like Carol was saying, I have a lot of experience working with plants. I don't call myself an expert per se of any particular group of plants. I think of myself as a generalist expert. I call myself a Jose of all plants, master of none. I grew up pushing lawnmowers in the LA area with my father, and now I'm up here in Northern California at the Botanical Conservatory, managing a broad diversity of plants. And I put this talk together again for I did it like on a chalkboard for our interns, and then I realized garden clubs could use this information because I love giving this talk actually to garden clubs in particular, because people have a lot of experience growing plants, but there's a good number of us that don't know the science of sort of how the hormones work. And so I think this will help understand why things do what they do, as opposed to when I would ask my father, why would he prune a tree this way and another plant that way? He would just say, because that's the way you do it. So I'm going to talk about how, what the science behind it. So real quick, I'm going to go through this real quick and review the parts of a plant kind of quickly. Normally I do this sort of a question and answer, kind of quizzing people, but since I'm just here talking, I'll go through it pretty quick. So the very tip of this right here, for those of you that are thinking, what is that? This is the growth point, or we call it the shoot apical meristem. It's the top of the plant. You're going to hear the word meristem a lot, and I'll sort of review what it means in more detail as I go along. Okay, so there are shoot apical meristems at the top of the plant. Okay, and I think you can see my mouse hovering over there. And then, Carol, can you see my mouse hovering over the top there? Someone's listening there? Okay.
Can I see what? The
okay.
arrow?
The mouse, yeah, the mouse hovering over.
Oh, yes, yes,
yeah,
I can.
Okay, good. I just want to make sure you guys can see that.
Yes.
And so there's the shoot apical meristems in the shoot system, the top of the plant. And then down in the root system, there are growing points at the tips of the active roots called, well, at the tips of roots, excuse me, they're called root apical meristems, okay? People that say these words a lot, they call them SAM and RAM, okay? But they call, the shoot tips and the root tips is another word, but I'm doing semi-technical here. And then, so you have the shoot apical meristems, and then you have the nodes, where a region on a stem where a leaf is connected. Okay, and right above the node, right at the node, okay, at the node, there's this concentration of meristematic tissue, and I'll go over that in a little bit more quickly. I'll just go over that in a little bit in more detail. So nodes are really important because they're the major sites of where new roots or shoots are made on a plant. And on a shoot, right above a leaf, there's this little bump right there, okay? And for those of you out there that are thinking, what is that? It's an axillary bud, okay? It's an axillary bud, and on a stem, that axillary bud is always meant to be a shoot, all right? It's going to grow into a new branch, and we're going to talk about the hormones that control that. But at the node, with hormone treatments, and well, as I'll talk about in more detail, you can actually get roots forming as well, and that's when we propagate plants. We take advantage of that. Okay, so an axillary bud is actually a dormant growth point. All right, it's actually the exact same thing as a shoot apical meristem, but the shoot apical meristem is active, and this one is just dormant. And we're going to find out about the hormones that control it in a positive and negative way. Okay, so what is a meristem? Well, I like to say it's the happiest part of the plant, the meristem. Haha. Thank you for laughing at my puns. Okay, but a meristem is actually a grouping of undifferentiated cells capable of being convinced to grow into something. All right, they're young. The cells are young. They're not mature. They can be directed to do this or that with hormones. Okay, whether naturally by the plant or in like tissue culture, for example. Okay. And we'll talk more about that as I go along, like I said. So, I always like to ask people, why do plants have meristems? What's the purpose of a meristem there? Okay, well, everything on the planet eats plants for the most part, okay? And so it's insurance to replace what will be damaged, all right? So it's really important for plants to have backup insurance, all right? I get frustrated over paying so much insurance, but here and there it's helped out, right? But I think of plants as the meristems and the plants as self-insured nonprofit organisms. Okay, so yeah, the axillary buds are insurance in case the top of the plant, the active growing parts of the plant, are damaged, so they can replace and take over. Okay, and we definitely take advantage of that when we propagate, trim, prune plants, etc. Okay, so. What is that section between the nodes called? Okay, and this is called the internode. All right, the internode, and the internode is basically a spacer between the nodes. All right, and something to note right here, an interesting little fact. John was talking about the Dreamia marittima, the sea squill, how it when he brought it indoors, it would elongate. Okay. Well, what's really interesting is that flower stalk, just like one of these internodes, and actually even a leaf, has a programmed set number of cells. They have a set number of cells that they make. But if you put them in the dark, they stretch out, okay? Or if you treat them with a hormone called gibberellin, that causes them to stretch out. And we'll talk about that when I get to gibberellin a little bit later, okay? Hopefully by the time I get to that, you won't think I'm talking a bunch of gibberish, all right? Anyways, okay, so let's talk about the two major plant hormones. And I apologize that normally when I do this, I'd sort of be quizzing you guys and going back and forth, but asking questions, but we'll do that at the end, and hopefully you guys are writing down or saving some questions to ask. Okay, but there's two major plant hormones, all right? And really the big one starts with an A. I'll give you guys a hint there. So those of you might be thinking, auxins, that's right, auxins, all right? And anyways, auxins are produced at active growing shoot meristems and young leaves, all right? If the leaves are fully expanded or the meristem is not active, then they're not producing auxin, okay? And like the axillary buds, for example, are not actively growing, so they're not producing it, but the growing points are, okay? And auxins, they travel towards the roots and with gravity, okay? So they try, because they travel towards the roots and with gravity, what part of the plant do they travel in? They travel in the sugar transport tissue known as the phloem, okay? And what's interesting is that on woody plants, on plants that get a little bit woody and big, the phloem is on the very outside of the plant, okay? So it's actually kind of sensitive, but usually it's protected by bark. But anyways, I digress. So it moves in the sugar, the auxin moves in the sugar transport tissue, okay? And, it turns out that sugars move from where they are produced to where they're needed. And so roots don't produce sugar. I see how they're white in my picture here. They're not photosynthesizing. They're sucking sugar from the top of the plant, and the auxin moves preferentially in that direction, all right? But we'll talk about anomalies or the things that can happen, alternative, okay? So auxins, as they travel down in the sugar transport tissue, and again with gravity, they inhibit axillary bud growth, all right? And it's because actually when at the top of the plant, they're in high concentrations, okay? So as they travel down, they tell these little axillary buds, don't worry about it, I got it all under control, okay? And so they tell those buds not to grow. But when they get to low concentration, especially in the roots, they actually promote root growth, all right? So they tell the roots to grow. And so if the top of your plant is very happy, actively growing, it's sending signals to the roots to grow more. And yes, it's so that they can grab water and nutrients to feed the top of the plant. We call that a positive feedback loop. So that more the top of the plant is growing and happy, the more it sends signals to the roots to grow. And then we'll talk about how the roots signal the top of the plant, okay? Auxins, we call them auxins because there are some natural there's auxin, a certain type of auxins in a plant, but what is an auxin? There's a bunch of different chemicals that can serve as auxins, and we traditionally use, when we propagate stuff, indole butyric acid, or sometimes naphthalene acetamide or indole acetic acid. There's a bunch of things that serve as an auxin that are a hormone that mimic that serve actually as an auxin, because what happens is that hormones, even in our bodies, have a certain chemical signal, okay? And in this case, there's a positively charged site and a negatively charged site on this molecule that is like 0.55 angstroms apart from each other, okay? I remember that from my development class like 35 years ago. But anyways, hormones typically have a certain chemical signature, and that's why in our human lives, we have to be careful about certain things like BPA and other chemicals that are hormone mimics, because they can disrupt our body functioning. And, they do disrupt plants functioning. For example, I believe it was Agent Orange that was used in the Vietnam War, was actually an auxin chemical that caused defoliation of trees, etc. okay? But let's get back to good positive plant growth type stuff, okay? So the auxins are produced at the active growing shoot meristems and young leaves. And then the other major plant hormone are cytokinins, and it's a much smaller class of chemicals. It's a very particular chemical, so there's not a lot of things that work as cytokinins, but there are a few different kinds. So auxins are produced up the top. Guess where the cytokinins are produced? Yep, you got it. Down at the root tips, okay? And it has to be active growing root tips, okay? If the roots aren't growing, they're not producing cytokinin, okay? And so that's where like making your roots happy is really important to making the top of the plant happy, okay? And we'll talk about that here as we go. So yes, oops, go back for a second. So cytokinins, excuse me, travel towards the shoots and leaves, all right? And so they sort of travel in the opposite direction, all right? You know, auxins travel towards the roots and with gravity, cytokinins travel towards the shoots and leaves, and they move in the water transport tissue, all right? So, you know, in contrast to the auxins that I mentioned that travel in the sugar transport tissue. So in the woody parts of the plant, the xylem, okay? And what's interesting is if a part of the plant has more leaves on it than another part of the plant, guess where the cytokinin is preferentially going to go? Yes, towards the area that has more leaves, because that's where the water is being pulled, okay? Cytokinins promote axillary bud growth, all right? So as they're traveling up the plant, they're telling the axillary meristems, the axillary buds, excuse me, to grow. Basically, feed me, Seymour, all right? I need more resources, give me more stuff. And so if the roots are growing quite a bit, then they're producing lots of cytokinin, and it tells the top of the plant to grow, okay? You can buy auxins are very easy to purchase because they're used a lot for a lot of different things. They're quite useful in a lot of different ways, okay, especially for propagation purposes. People don't buy generally as much cytokinin to use for things, and so it's actually kind of hard to get, but thanks to Amazon.com, you can buy some of it these days. And you can get it in this, it used to be in lanolin. Nowadays, it's probably a cheaper product, a petroleum product, but they call it keiki paste, okay? And you may have heard on Phalaenopsis orchids, the keikis, the little plantlets that grow sometimes on the flowering stalk. That's because when a plant is overdosed with cytokinin, it will grow vegetative and make vegetative parts. And I'll cover that more as I go along with a few other slides. Okay. And I apologize. This is where I would take lots of questions, but we'll cover things as we go along, and I'll try to sort of let's see. I guess guess some questions that might be asked out there. Okay. So what happens when you prune plants? Okay, so let's look at this. Here's my sort of schematic plant again. You know, and this could be a salvia, it could be a, I don't know, the sea squill maybe, or some some kind of all sorts of plants basically work the same way with the hormones. Okay, so let's talk about pruning a plant or cutting a black a plant, because we're going to talk about the roots and we don't talk about pruning roots too much, but let's talk about this. So in this case, I'm removing the top of the plant right above that little black mark. I'm removing that part. And now in this situation, oh, and by the way, before I get to that, I'm going to talk about, I just want to point out, when I use my arrows over here, and I'm pointing at the screen, but you can't see me pointing. When I use the arrow up like this, it means high amount, and I use the arrow down, it means low amount, okay? So in this situation, so we can review, we've cut off the top of the plant. So what hormone have we cut off from the rest of the plant? Yes, auxin, okay? And we're still gonna have cytokinin because that's produced at the roots. So in this situation, you have low amounts of auxin right in here, Okay, this is what we care about. We cut off the top of the plant. We care about this area right here. And we have low amounts of auxin and high amounts of cytokinin. Okay, and that equals shoot growth. Okay, I just mentioned earlier, when you have lots of cytokinin, you get shoot growth. And by the way, if your plant, if you're over fertilizing your plant and you have a very vigorous root system, the plant isn't normally very happy, you're going to get much more vegetative growth and you're not going to get as much flowering. Okay, and that's something farmers have to be careful about, if fruit trees, etc., of not over fertilizing them and getting them to be vegetative instead of flowery. So you want to be careful not to overdose the top of the plant with cytokinin. Okay, so yeah, oftentimes you will get one single axillary bud breaking dormancy, but in plants that are a little less sensitive to auxin and very reactive to cytokinin, you will get many more multiple branches growing out of there. I like to think of like a eucalyptus tree. That thing gets cut, and I'm not a fan of eucalyptus, but that thing gets cut, a bunch of shoots will grow out of there. Okay. All right. So yes, low amounts of auxin and high amounts of cytokinin equals shoot growth. And by the way, when people do tissue culture, and they tissue culture a part of a plant, and they find a part of a plant that has some amount of meristematic tissue in there, they culture it, keep it sterile, and then they treat it with this combination of hormones, so you get shoots forming. Okay, and it's typically done in a very enclosed environment, so you don't have to worry about it drying out. But then later on, they switch and they use more cytokinin, more auxin and less cytokinin. And let's look at that here. Okay, so oh, before I get to that, excuse me, I forgot this part. So some of you out there may have cut some roots on a decently sized plant sometime, and then you were shocked to have this happen. Shoots come right out of the ground. Okay, it happened with me to me with a pear tree, a fruitless pear. I cut some roots for some irrigation I was putting in somewhere, and then I had these shoots popping up out of the ground, and I thought, what's going on? And then I was like, wait a minute, hormones. Okay, in this case, right in there where I cut that root off, separated it from the rest of the plant, cytokinin was being produced at these active growing roots because they still had some energy to them, and they sent that cytokinin up, and there was a lack of auxin, and boom, you got shoot growth out of there. Okay, and when that usually happens, you think, shoot, that's a problem. Sorry, I had to do it. Okay, okay. Here's the other situation when you transplant a plant, replant it. You cut off in this case, we're cutting off the lower part of the plant. Okay, and I haven't figured out in PowerPoint how to make this disappear really well. But anyways, imagine this being taken away. And so in this case, we care about this little section right here above it. And right here in this situation, you have auxin, right? That's traveled down and built up right there, but you don't have any cytokinin. And so you're going to get roots forming, okay? And that's because in this situation, you have high amounts of auxin and low amounts of cytokinin, and that equals roots, okay? And this is really important when we're propagating plants and making cuttings of them, because we cut right below a node, okay, when we're making a cutting, cut right below a node, and then you have to put it in the right environment. And I'll have a quick little video of how we propagate plants. But you cut it right below the node, and in this situation, right in here at the node, okay, you're going to get a buildup of auxin, right, just like you did right here, and a lack of cytokinin, and you're going to get roots, okay, if you put it in the dark and keep it moist, etc. Okay, if it's succulent, it might happen just sitting on a bench top, okay. But yeah, you're gonna get roots growing out of there. Okay. Yeah. So and by the way, I guess this is what I wanted to point out here, is it's really important when you're pruning plants to prune right above the node, cut right above it, so that when you get your cytokinin traveling up and it builds up right at the node, not with excuse me, with in the internode. You don't care about the internode is just a spacer. You don't want that. Okay. So and then when you're making cuttings for rooting purposes, you cut right below the node so you maintain the meristem. Okay. The node with the meristematic tissue is really important when you're pruning, and when you're propagating, okay? It's all about the node. The internode is just the spacer, okay? All right, so I like to use this example because most of us have grown tomatoes at one time or another. And, you know, I never knew why you did this, but I just put it together myself after, you know, processing, not tomatoes, but processing hormones in my head. And so, you know, I always wondered, people talk about how you're supposed to bury a tomato plant deeper when you plant it. And I don't know if you guys, some of you guys have done this, you know, or you haven't done it, but they talk about doing this, and I'm going to tell you why. It's really cool to me, okay? Hormonally, how this works. So you take a tomato and you strip the leaves off of it because you're going to bury it. And then you take it and you take it out of the pot and you soften up the root system, loosen up the roots so that they get into the new soil. It's a good transplanting technique. You just want to be minimal damage, but you want to get the roots out into the new soil. And then and you get, tomatoes are really tough, so you can almost do whatever you want. But then you take and you bury the tomato plant. And you know, like when I first heard of this, I was like, what? You're taking away the photosynthetic potential of a plant. And you know, what are you doing? What's going on? Well, by burying the tomato plant, you have all this stem here with meristematic tissue at the nodes, and because it's actually, tomatoes are kind of herbaceous, they actually have meristem, a pretty good amount of meristems, meristematic tissue along the whole stem, and you're going to get a bunch of roots forming on there, what we call adventitious roots. And so, before I tell you the answer, what hormone is produced at all those active growing root tips? Okay, yes, you got it, it's cytokinin. And with all that cytokinin that's being generated, okay, out those roots and traveling upwards, along with water and nutrients, okay, you're going to have, you're going to generate lots of cytokinin, from all the new root growth, because they're actively growing those roots, okay, because the stem's buried, and anyways, you get a lot of new root growth, and that means you're going to get a decent amount of shoot growth. In fact, more than if you had just taken that tomato and replanted it into a bigger pot and not buried it, because in this situation, by generating more roots, what you're doing is you're causing a hormone imbalance, okay? And because you're rebalancing the plant to have lots of cytokinin in the top of the plant and very little auxin, relatively speaking, you get a bunch of shoots growing out of there, and you've actually sped up the growth of the plant with hormone therapy, okay? So in this case right here, I want to take a little break to talk about, let's see, what else do I have here? Oh yes, so by the way, this burying the plant right here is also a good way of propagating things like mints, geraniums, basically anything in the mint family, many things in the mint family. You know, you can bury stems and they'll form a good amount of roots on them, okay? And other things like Christmas cactus, etc. All right, but what I want to point out right here is when you're pruning plants, okay? So those of you guys who are in Southern California, and I grew up down there, and I saw tree pruners come in, and the tree was kind of overgrown, and then what do? They prune it all at once, a lot. All right, so they removed a bunch of shoots on the top of the plant, but did they go and root prune the plant? No. And so what happens to that tree that's been pruned heavily? It has a vigorous regrowth, and then it's going to need pruning again at some point. And so, if you think about like people that do bonsai, what do? They prune the top of the plant a little bit, okay, to minimize the active growing shoots, okay? And they also constrict the roots so that those aren't producing what? Cytokinin, right? So they constrict their plants so they're not producing lots of cytokinin, so you don't get vigorous growth on the top of the plant. And then they keep the plant pruned back at the top to really control and direct the growth, but put the plant almost in like suspended animation to slow the plant down, okay? And that's what they do in bonsai. And so what I do nowadays when I prune my shrubs at home, or I do some weekend work, doing landscaping, etc., pruning it for people, I come in and I prune a little bit at a time, all right? and then I wait a few weeks, I come back and do a little bit more. And at home, if you do that, you won't have these repulses of growth, okay? Because overgrowth is repulsive, right? Okay, anyway, sorry. So at the conservatory, we used to pull our plants into our workroom in the middle of summertime when it wasn't very active. And we would take them and we would prune them back because they were kind of overgrown in the greenhouse. And then we would say, oh, let's go ahead and transplant them at the same time. And what were we doing? We were removing the auxin generating parts at the top of the plant. And we were also removing the cytokinin generating parts of the plant. And we were putting the plants into suspended animation, but not like bonsai people do, which they look pretty cool. We were actually stunting the plants by getting rid of both control points, okay? And so it's really important when you're transplanting to have one part of the plant be active or the other, okay? And this is why when we transplant, you want to transplant things when they're actively growing or just before they're growing, so that they regrow, okay? And we could talk more about that in the Q&A part, okay? Really quick, people did some research to figure out how the hormones work, and they figured out that it wasn't the leaves themselves per se, but it was the auxin that was in the leaves. So a young growing shoot right here, is producing the auxin. But if you strip the stem off and without any leaves and meristem, no root growth. But if you add auxin to this bare stem, you actually get root growth, because it's actually the auxins, not the photosynthesis that's causing the roots to grow. Okay. All right. Yeah. So and I'll cover some more things as I go along. But so again, if you guys are making cuttings, and I'm sure a fair number of you like to propagate plants, or I'd encourage you guys to try it. Okay. When you take cuttings of a plant, again, it's really important to have young, active growing leaves and active meristems, so that you get root growth. Okay, we go in and we add supplemental auxin to the cut end of a plant. And by the way, if I was cutting this, I would actually cut straight across, especially for an opposite leaf plant. Okay, we add a little bit of hormone to convince the tissue right there. As soon as we cut it, you put it on there to direct it to form, to start being convinced to grow roots by having the high amounts of auxin and no cytokinin. Okay, but a number of years ago, my colleague was moving from one house to the other and had a bunch of rose bushes in the middle of summertime. It's a 100-degree day, and she asked me to take some cuttings of her roses, and I went through her shrubs, and I went fishing around for the one or two branches that had active growing shoot meristems and young leaves, so that I knew that when I took that cutting, I had a continuous source of auxin. And I'm proud to say that every single rose cutting that I took on that 100-degree day, and those roses were already stressed for water, all of the cuttings took because I went and found the ones that active growing meristems. Okay, as opposed to that one time where I went to a botanic garden and I spent some time with someone who was a plant collector, not a horticulturalist, and he was giving me that little side branch down below on the side of the plant that wasn't very active, wasn't producing auxin, didn't have young expanding leaves, and I barely had anything that rooted from those like 40 different species of cuttings that he gave me. And I'm a botanic gardener. I spent time entering into the database, and a lot of it was for naught, because the cuttings were not good cuttings. They were those branches on the plant that are on their way to going permanently dormant. Okay, so that's why growers take their cuttings from the top of the plant. That's where you want them from. Okay, yeah. So that's where I learned my lesson there. Okay. By the way, if any of you are into succulents, okay, succulents and slow-growing plants are very sensitive to being overdosed by hormones. And in fact, in general, you want to be careful not to overdo it, because if you add too much hormone, too much auxin will actually stunt growth, like I was talking about earlier. Okay, so when we do succulents, we use low active hormone and an auxin that's not very active, like NAA, naphthalene acetamide, or naphthalene dicrylamide. These are low active, and sometimes it used to be, it's harder to get it these days. It had some fungicide in it, which is great for succulents to keep them from rotting. Okay, so important thing to note here when you're making cuttings. Okay, remember I talked about how auxin travels with gravity. All right, whenever you make cuttings, you want to keep them upright. Okay, because you want that auxin to travel all the way down and build up at the bottom there, so that you get roots forming down here. Okay, if you have your cutting laying sideways, then the auxin, especially if it's really flat, the auxin is going to pull up along the whole thing and get diluted, and you're not going to get as quick rooting as if you get it to build up at the very bottom with gravity. Okay, all right. When you have what are called softwood cuttings, okay, green herbaceous material, okay, you use low amounts of auxin because there's lots of meristematic tissue there that's easily convinced. As you get into what semi-hardwood or hardwood, you start increasing your amount of hormone. And when we do woody cuttings, things like pines and things that are pretty woody, you use higher amounts of the hormone here. When I do like ginkgo cuttings, I use these higher concentrations of hormones, because in here, in a young plant, herbaceous plant, you have lots of meristematic cells or potentially meristematic cells, undifferentiated cells in between the vascular tissue here. Whereas when you have more mature tissue, you have your wood that's all very differentiated, and then you have your phloem on the outside, but you just have that one little sliver of what's called the vascular cambium. And you can see right here, this is showing a little drawing of a shoot growing out of there, and this is roots, okay? So this could be a woody plant, and like when people top trees, you get these shoots growing right from the vascular cambium, okay? And we could talk about why that's not good, because they grow really vigorously, they're weakly connected, etc. But that's a topic for another day, okay? This is just shows it in sort of like a little more detail, how it's more herbaceous up at the top and green, and it gets woodier down below. But you won't be tested on this, okay? I want to show you this because, you know, again, the importance of young leaves and meristems for better root production. So a number of years ago, I was teaching our interns how to make cuttings. And by the way, what I do is I do the hormones lecture 1 week, and then the next week we propagate, so it's fresh in their minds, okay? But the student had forgotten some of the details of what I had told her, okay? And so you see this cutting on our right here. You see the young leaves in various places, young expanding leaves, okay? And it's actually got multiple, it's got, this is a shoot right here, and this is the main shoot over here. Whereas this one over here, you don't see any signs of young leaves on it. And I'm sorry, they're a little crowded together, but there's no active meristem on here. And look at how much less roots we got from the cutting that wasn't very active versus the one that had active growing shoot meristems and young leaves. It's pretty obvious how that works, okay? Yeah. Oh, let's go back for a second. And again, it's really important when you do take cuttings from things that you typically do it when the plants are actively growing, okay? Because they're going to be more reactive. They're going to, you know, because they're actively growing, they're going to generate the continuous source of auxin after you do your initial applications, all right? Yeah. So I'm going to show you guys this quick little video here, if I can play it, and you might get the sound coming out of there, but if not, I'll narrate it, okay? This is how we actually make a cutting. Oops, go back for a second. So this is what we use, yeah. You guys can hear the sound?
I'm not hearing it.
No? Okay. So sorry. Thank you. So in this case, I'm stripping off the leaves there. I had wet vermiculite perlite. Okay. And so I have pointing out that this cutting has young, tender, expanding leaves on there. All right. So now I'm going to take my pruner and make a fresh cut. Get the hormone out of there. And by the way, when I use the dust form, I like to roll it out so I don't generate too much dust and don't breathe it in. Here we go. I'm cutting it right below node. Boom, quick cut so you get a smooth, good cut. Dab it with the hormone. Knock the extra off. Poke a hole so I don't rub the hormone off when I stick it in there. Sit in there. Pack it in tight. Okay. And then, yeah, so this is I'm going to demonstrate another one right here. But what I would do here, once I do this cutting, is at the conservatory, we'll take it and put it on a propagation bench with mist so that it keeps it moist. Here I'm cutting at the node again. Talk about how you typically want cuttings that are 4 to 5 inches in size so they're not too top heavy and fall over. Same technique there. Poke it in again. And then at home, what you can do is do this, and then you can put it into like a Ziploc bag that I have in the background, or somewhere in shady, cool situation. Yeah, so a little bag right there. All right, so we could stop right there. So yeah, we put it, you know, at home you could do this into a Ziploc bag because you want to keep it humid because these are leafy plants that would dry out. If it was a succulent, I would be doing it into pumice or gravel in the open. But in this case, for cuttings, you know, things that are leafy, they would dry out. So we do this technique. And by the way, this video, this whole lecture is available with more details via our website, and which I'll have to send the link to the group so that you guys can download it, because the intermediary location is down at the moment. Okay, but let's move on. Hopefully, I haven't made your eyes roll, you know, in the back of your head too much with all the details here. But anyways, let's continue. So you may have heard of air layering before, okay, or layering, and you know, this is a technique where what you do is you go in and you strip the phloem of the plant off. You strip the outside of the plant, the phloem, you leave the woody tissue. And what that does is now auxin, again, is going to travel down and build up right here. And you can get roots if you keep it humid and moist, like when we buried the cutting. So in this case, what we do is we wrap it with some kind of material, in this case, sphagnum or rock wool, something that's moist and spongy. You wrap it with plastic to keep it from drying out. And then what I do is I could wrap it with aluminum foil outside of that. So we'll actually look at that here. So this is actually used to clonally propagate difficult to root woody plants, okay? And so here's the technique, stripping the bark off of this plant right here. I forget which plant this is, but stripping the outside so you get down to just the wood. Dabbing on a little bit hormone at the bottom of the stem that we want to root. So we're only putting it right here because that's where the meristematic cells are, okay? So there's the hormone. We're using low amounts of hormone. Okay, wrapping with sphagnum. There's the plastic right there, tying it in so it doesn't dry out. Okay, and then wrapping it with aluminum foil to make it dark because roots don't like it. Most roots don't like sunlight. Okay, and so that's how you do something called air layering. There's also a technique called layering where you just take a branch and you lay it sideways. I'm trying to do my arm here. You lay it sideways, and when you do a branch like this and there's a dip, okay, auxin is going to build up right there. And if you take that, it's touching soil or it's buried, you'll get roots growing out of there because auxin pulls up, and that's going to cause roots to grow. Okay. Yeah. All right. I want to speed things up so I make sure I get to the other hormones. So really quick, this is a comment. If you see the plant on the left here, I call this plant a shrub, and I call this plant a tree. Okay. The one on the right is very apically dominant. The top of the plant tells the little meristems at the base of this bulb not to grow. Whereas this plant over here is a little more like a shrub. It's a little less sensitive, so you're getting much more branching. Okay, and that is really the difference between a tree and a shrub, is that a tree is very apically dominant, very auxin controlled, and a shrub is less sensitive to auxin, and so it's more branchy because it reacts to the cytokinin more. Okay. And I had the situation where I was overwatering this bulb trying to get it to grow faster, and I actually rotted out the mama plant, okay? but luckily I didn't kill off my whole bulb, and I actually, by taking away the main bulb here, the little axillary buds down below grew out into new bulbs, okay? I like in this situation over here, the helicopter parenting, that don't let their kids mature. Ooh, sorry, I said that. Okay, all right. Really quick, splitting the meristem of an aloe to get it to branch, okay? This is actually really cool. People do this all the time. Just gonna speed through here. We actually tried adding cytokinin to get it to branch more, and this aloe is so slow growing, this rare aloe, Aloe palanzii, that I actually stunted it when I actually used the hormone, so it's better to go natural. All right, and it turns out you can split meristems on plants like succulents that have big meristems because the apical meristem is made up of many cells, and if you split them apart from each other, okay, by like quartering it, then you have 4 quadrants and they can all grow on their own as if they were separate. And look at this aloe, look at the aloe where we did this here, okay? 1, 2, 3, and one of them became the dominant one because that's always happens with apically dominant plants. This one over here, I lost the other 2 because those are the ones that I treated with cytokinin. So I learned my lesson in this case to just split these and not use any hormone, supplemental hormone, okay? Don't use unsterilized dull tools because they cause a lot of damage. So I lost this one because of that. There it is more mature. And by the way, I've done this with cacti. I talked to a Plumeria society about doing it with their plumerias as well. Okay. I'm going to sort of skip through this, but just really quick. When you graft cacti, in this case, we took a cactus and we grafted it onto a cactus with a more vigorous root system, and that caused it to branch a bunch because it got lots of cytokinin and it caused it to branch. Okay. So I want to skip through, so I want to get to the other hormones, but yeah, I'm going to skip this part here. Okay, so these are all the functions of auxins, okay? You won't be tested on this, but the website up there is where you can look this up to review some of this information in a little more detail. And if there's questions at the end, we can jump back to this slide here, okay? Cytokines don't need nearly as much, all right? But I want to finish up and talk about the other 2 major hormones in plants, okay? And the other one I mentioned earlier is our gibberellins, okay? And I sort of touched on them a little bit earlier on, but gibberellins are interesting because they're produced throughout the plant, okay? Whereas like auxins are only produced at active shoot meristems, and then cytokinins are produced at active root meristems, gibberellins are produced throughout the plant. But they're broken down by sunlight, okay? And what they do is gibberellins promote cell elongation and stretching, all right? So if we think about that sea squill again that John was talking about earlier on, or I'm thinking like table grapes. What do they do with table grapes? They actually spray them with gibberellins to get them to swell up and expand, okay? And that causes the cells to get bigger. There's a set number of cells in that fruit, but they add gibberellin, and those cells swell up and get bigger, and so you get a bigger grape, more diluted, but hey, it looks bigger, someone will buy it, okay? In contrast, if you guys heard of poinsettia plants, people buy those poinsettia plants and they're really nice and compact. They take them home, and if they keep them alive, after a year or 2, they stretch out and they get really big. Well, they treat them with a hormone an anti-hormone, hormone inhibitor called bonsai. It inhibits GA synthesis in the plant. So even if you have your plant growing in the greenhouse in the shade, it'll still stay compact because the gibberellin doesn't cause the cells to stretch out, okay? And if you think of plants in shade versus the sun, leaves are bigger in the shade than they are in the sun. That's because in the shade, the gibberellin doesn't get broken down as much, and the cells stretch out and get bigger, all right? And your internodes stretch out and get bigger, all right? And that's again with that plant being brought indoors where it gets stretchy, okay? So it's kind of cool, all right? But again, the growers will treat plants, a lot of house plants, with these GA inhibitors to keep them nice and compact so that they look really good. And then you try to grow them and they're like, yeah, I don't do that naturally. Okay, let's finish up with the with ethylene here. All right, hopefully I haven't blown a lot of gas in your direction here, hot air or whatever. Haha. Okay, so ethylene is called the ripening hormone. Okay, ethylene promotes ripening and cell maturation and death. So it causes cells to kind of do their final thing and get bigger. If you've heard of bananas, they pick them in South America, here in North America. They pick them in South America and tropical areas. They pick them kind of green. They put them in these airtight boxcars so that they don't rot in there. And then when they do somewhere along the way, they gas them with ethylene, and that causes the bananas to start ripening, okay? You also may have heard the saying, one bad apple ruins the lot, okay? Because an apple that's maturing in a fruit basket produces lots of ethylene, and once you start that ethylene process, you can't stop it. And if that apple ripens quite a bit, it'll cause the other apples to ripen, or like what happened in a box of peaches to me the other day, okay? So what's really cool with plant growth is this may have happened to you, is it, and I'll be finishing up in like 2 minutes here, so we have time for questions, okay? What happens, you may have had this happen where you brought a plant from a nursery, you brought it home, or you moved it from one place to the other, one place in the house to another, et cetera. And then you had a bunch of leaves turn yellow and fall off, okay? Well, it's actually really cool how this works. So, auxin travels with gravity and towards the roots where the sugar is being sucked. Well, if you move a plant and it can't photosynthesize this sufficiently to make sugar, it will actually start sucking sugar from the rest of the plant. And when you start pulling in sugar into a leaf, that causes auxin buildup. And high amounts of auxin trigger ethylene production, and that causes the leaf to turn yellow and fall off. I call it the anti-mucho mechanism. It keeps leaves from sucking off the plant or any part of the plant taking too much, okay? It actually has to be, the top of the plant has to generate sugar for the plant, not take it away, okay? So we can talk about that more with the questions. So, this is what happens with fall color, okay? In the fall, and this is in the desert where it's drying down, the leaves of the plant are stressed out, they can't photosynthesize, they start sucking sugar from the top of the plant doesn't want those leaves there, so they don't suck water out of the plant, and because there isn't enough water, so the leaves start building up auxin because they start pulling in sugar instead of making sugar because they can't photosynthesize, and they turn yellow and fall off, okay? And it happens very consistently on the East Coast because the weather all of a sudden switches around Labor Day or whatever to kind of colder weather. And is that right? Labor Day, I can't remember the holiday, in the fall. And the leaves can't photosynthesize, so then they start turning yellow and falling off, okay? So yeah, this is Ocotillo, happening in Ocotillo. So these are some of the things that gibberellins do, okay? And they're actually, it's important to be for seedless fruits. Fruits typically don't develop if they don't have seeds in them because the seeds in the fruit are a source of gibberellin, but people use it in seedless plants by adding it, okay? Yeah, and actually, and I go back for a second. I forgot to talk about this real quick. Gibberellins are important in seed germination because the seeds, those cells start expanding and stretching, and gibberellins assist in that, okay? And then, here ethylene, all right? And again, the important thing is it causes senescence and fruit ripening. And it's actually interesting in this right here, this line right here, induction of femaleness in dioecious flowers. So you can actually get plants to switch from male to female if you use ethylene in the right way. So lastly, before we get to the question and answer session, I want to point out, so here's a happy plant in the desert, okay? And this plant is huge, okay? And it got huge because it had water and nutrients to it, all right? The plant over here in a rocky area is more sort of bonsai'd because it had less water and nutrients available to it. But what caused the shape of the plant, the less branching, where the more branching on the right? Hormones, okay? Hormones direct the growth of the plant. So they're the direction, but water and nutrients are the fuel, okay? and sugar. All right, thank you very much. I'll stop there and we could work on questions, okay?
Thank you, Ernesto. That was a super fascinating presentation. And thank you everyone for sticking through with it with us and me with my crazy internet connection. I hope it wasn't too distracting. Before we move on to the Q&A, I do want to just make a quick announcement that our meeting next month is actually on a special day. It's on Thursday, September 24th at 7:30 p.m. It will be a Zoom webinar. However, this meeting is special because it's actually our Horticulturist of the Year award ceremony. So again, this one is going to be a little bit different because we're not necessarily having all of our attendees present with us. So stay tuned for more details because it is going to be a great night honoring some very important horticulturists that have helped Southern California and just the world in general, okay? And now, before we go to the Q&A, I know some people may have to go, but I do want to get to our raffle, which a lot of people love this raffle. It's really fun. Ernesto, you're gonna see our wheel of names right now. So I'm gonna share my screen.
Oh, should I unshare? Should I stop sharing?
Oh, I can. I'm gonna share over you. Don't worry.
Okay.
All right. And as you see our wheel of names, we're going to tap to spin and see who our lucky winner is. Katie B! Katie B, if you are here, please raise your hand on the Zoom screen. Let's see, let's wait a minute to see if Katie B is here. I don't think I see her. Oh, Katie, you're here. Awesome. Congratulations. You are actually winning a copy of Plant Propagation, the 8th edition by Hartman and Kester. It is one that Ernesto suggested, so it's sure to have plenty of information for you. And we are going to stop this. Congratulations. We'll be reaching out to you in an email after the webinar, okay? So let's get started with our Q&A then. Ernesto, I will be reading them off to you, so feel free to answer as they come, okay?
Okay.
First one is from Kathy C. She says, where were you in my science classes? This information, this is great information. And then she goes on to ask, during the video, I noticed that you left most of the leaves on the stem of the cutting that was planted. I have been rooting cuttings and I always remove all but the tiniest leaves on the very tip. Is this wrong? Would it be better if I left the leaves? I'm propagating. Oh, keep going.
Yeah, thank you very much for asking that question. It's a really good question because when you make a cutting, what you want is all the young expanding leaves. So I could have removed a few more leaves on those plants, but those plants are tough and easy to root. But if it's a more sensitive plant, I would cut off all the leaves except for all the ones that are young and expanding. Those are the ones that you want when you're making a cutting. Yeah, really good question.
Awesome. This one is from Sienna C, who's actually on another time zone right now. She says, is this burying technique affected by the climate of the local area? I'm considering how viable it is where I am in the UK with a climate colder than California's.
Well, it depends on the plant. The burying technique will work on a fair number of plants if you do it at the right time of year when the temperatures are good, typically on the warmer side. So I wouldn't do it in the middle of winter time. I might do it in spring or during the summertime there in the UK where you're taking the stem and you're taking it and burying it, right? Yeah. That's all I could say about that, I think.
All right, thank you.
Yeah.
The next one is from Theo F. What is the time frame on keeping cuttings vertical? Would it matter much to have them horizontal for like 15 minutes while processing?
Oh yeah, no, I mean, plants don't move that fast, but I would say by the day, but few hours into it, it'd be good to keep them upright. In fact, when I do succulent cuttings, which they don't dry out on me, and I want the bottoms to heal so that they don't rot out, I will actually stick them in a pot and sit them upright. Okay, So I would say in a matter of a few hours, you want them to be upright so that the hormones are traveling down.
All right, thank you. The next one is from Patricia S. What causes the axillary buds along the portion of the stem of the tomato plant to switch development from lateral branches to form adventitious roots when buried in the soil? Is it because the associated leaves are removed or destroyed, which in turn creates a phytohormonal imbalance?
Well, so it turns out that I mentioned it earlier a little bit, is that along that stem, you know, you had your leaves that were removed, and there's nodes there, and at the nodes you had axillary buds that there were definitely going to be shoots. Okay, sometimes you'll get shoots coming out of the ground from the tomato from those nodes, but all along the rest of that stem of the tomato is just meristematic cells that will then form the roots. So the roots are formed at and around the nodes along the whole internode. Okay. Yeah, so they form multiple places. Yeah.
All right. The next one is from Elise M. When using a cutting for propagation, if I'm not putting it in the vermiculite right away, should I keep it in water? And apologies for butchering any of these words.
Yeah, it's actually really important when you make a cutting is to not let that bottom dry out too much. So you put the hormone in there, and then you want to put it in the media right away. And like you're saying, if you're gonna do a pretty much if you're gonna sort of somehow do it in stages, yes, setting them in water so that they don't dry out is really good. You want to treat them kind of like a cut flower. You know, when we bury them in the vermiculite perlite, it's really humid down there. That cut end can still absorb water and feed the top of the plant a little bit.
All right, thank you. The next one is from David N. Do you recommend hormone treatments to deal with suckering from plants like crepe myrtle?
Actually, yeah, that actually is, and you can do it fairly organically. You can get some kind of compound that's an auxin compound, and if you put enough of it on there, you should be able to inhibit was the suckering. And in fact, one thing I didn't cover in the talk is that when you have a stem and it's laying sideways, the auxin is going to travel on the bottom right here, and that's what happens in the crepe myrtle. There's some roots near the surface, and they don't have any auxin up here, so you start getting shoots growing out of the top of that. Mm-hmm.
All right, the next one is from Yahir G. Is Michelia alba complicated to propagate in your personal experience?
We've propagated Michelia champaca at the conservatory. It's sort of a woody plant. It's a little tough to grow, to get to root, excuse me, but you know, putting it in vermiculite perlite and then on a mist bench or somewhere where it's cool and not so bright, no direct sunlight, you can get them to root. And I think from what I remember, we used the Hormex 16, the 1.6% IBA compound.
All right, thank you. The next one is from Abdulaziz A. What is the difference between rooting hormone and cutting hormone?
Oh, it's the same thing. Yeah, yeah, it's the same thing. You typically call it rooting hormone, but I suppose that out there some people have sold it or published it as cutting hormone, but it's the same thing.
Oh, that's easy.
Yeah, it's
right.
auxins. Yeah. Mm-hmm.
The next one is from Elaine. I have just taken some large sedum cuttings. How moist does the potting media need to be kept?
Okay, good question. Sedum is somewhat of a succulent. So, I mean, it's a succulent. They're not a super drought tolerant succulent. It actually depends which sedum it is. But my suggestion would be is to take those stems, strip off the parts that you're going to bury, and then put them upright in some very porous media, like pumice or some fine gravel, even like sifted decomposed granite if you're taking out all the fine stuff. So something very porous, so that it can be humid, dark, but lots of oxygen so you get roots and no rot. Yeah.
All right, thank you. The next one is from Portia. Do you remove leaves and things when you pot things into their own pots after the cutting has rooted?
No, not typically. Yeah, no, I mean, we remove old dying leaves off of it from the cuttings, but otherwise, I mean, really, you could remove the older leaves if you're worried about your cuttings drying out, okay? But once they're rooted, they should be fairly easy to feed the top of the plant because you have lots of roots for the amount of shoot system that you have. But you could strip off the mature leaves and only leave the young expanding leaves so you keep continuing promoting growth.
All right, the next one is from Laura B. Why are some auxiliary buds so hard to get started? Thinking about hard pruning on plants like lavender or California sages in which buds won't break on older wood.
Ah, okay. What's happened, I thought about this, what's happening on some of those things like lavender is that there's very little meristematic cells left in those very old woody stems, okay? There's some, but there's very little, and so that's why you don't get growth because there aren't any meristems. So like on lavenders, that's why we talk about only pruning to where there's still some green tissue so that you get bud break, because there's not going to be any meristems left in this very woody tissue. It's like an oak tree. I don't think like oaks really will root. I mean, some of them will, but anyways, it's a very woody, all the cells are very mature, so there's not much meristems left down there. Okay.
All right, thank you. The next one is from Carol A. Are roots that grow in just water still viable? In essence, an avocado root in a jar.
Oh, I mean, yeah, they're I mean, they're roots just like roots in soil. When you do them in water, you want to be careful when you transition them to soil because they can kind of dry out. But yeah, they're the same thing. They're just, it might be a little morphologically different because of the very wet environment. But then good luck having that avocado be a fruit producing avocado. Okay.
The next one, and I apologize in advance if I don't say your name correctly, Isult B. I did not quite get at the beginning when you cut roots, why one was growing shoots and the other one grew roots.
Oh, okay. Because when I was doing that I was talking about, when I was cutting about shoots, I cut a root and I was talking about the root that was left behind where there was only cytokinin and no auxin. All right. So in that case, the root that was cut off from the main plant had cytokinin and no auxin, so you got shoot growth. But now you have a root coming from the top of the plant and I cut it off. Well, right above here, I'm going to get root growth because I cut off the cytokinin, cut off the rest of the root. So I was talking about one side of the cut versus the other side of the cut. Okay. Yeah.
And this is actually a great point because I'll make a note that tomorrow we will have this recording on our YouTube channel. So if you missed any part of it, any part of Ernesto's presentation, we will have it online as well. And Ernesto will send you that link too.
Great.
All right, we have another international viewer, Amanda H. I'm here in Australia and I propagate frequently and also do workshops too, here for new and experienced gardeners. One thing that you didn't touch on is how many nodes do you plant into the mix and does it vary depending on species? Generally, I say 2 under and 2 or more above. What do you do? Also, I callus off most succulents before planting.
Yeah, it's definitely very important to callus off succulents before planting because you can get rotting, okay? So I callus them for 2 weeks to 2 months before I bury them and sitting upright. But yes, the question of how many nodes, it really depends on the plant, okay? When I do most cuttings, 90% of the cuttings that I do are 4 to 6 inches with, like she was saying, at least 2 nodes buried under the soil. 1 node is fine for some things, but 4 to 6 inches cuttings with half of it buried and then half of it above, okay? I do capers, and I found out with capers, when I do cuttings, I use like 2-inch cuttings of very young green tissue, and that roots much better than semi-hardwood material. So it really depends on the plant. That's where looking up, techniques of how people have done it before really helps out. Yeah.
Right, here is one from Caroline B. I'm having trouble with Salvia canariensis cuttings. Any hints?
I'm not familiar with that plant, but I would suggest trying them whenever they're sort of actively growing. And when you take those cuttings, I think that one kind of crawls sideways, but I would still take the stems and bury them upright. And again, whenever I do cuttings, I put them you can put them in bright light, but it has to be indirect light, so you don't get any light cooking on the plants. And if the plants can tolerate some amount of shade when they grow, then definitely have the cuttings in shady conditions so you don't get drying out before you get rooting.
All right, the next one is from Brad H. Where are the auxins produced on a leafless plant, such as a columnar cactus? Excuse me.
They're produced at the apical meristem, and so those succulents are producing less auxin than the things with leaves on them, and so that's part of why they grow slower. Okay.
All right, we have another question from Sienna C. At school, I was taught that auxins promote cell elongation, especially in darkness. Were they actually gibberellins?
That's a really good question. And one thing that I kind of sort of didn't really talk about, it brushed over, is the fact that these hormones interact with each other. Okay. And when you have a stem and it's laying sideways, you get bending back up because auxins stimulate cell elongation in there in low amounts because it's moving through, so it's not hanging out there. So it stimulates cell elongation. So that is true. It does do that. Okay. Gibberellin plays in there a little bit, but that's more of a light shade situation. But yes, auxins in low concentrations, they promote cell elongation, and in high amounts, they inhibit cell elongation, and that's why you don't get blood break. Okay. Yeah.
Here's another question from Kathy C. Question on plant hormones in children. I gave someone some cuttings and plant hormone to work with her 9-year-old. I was concerned because I didn't know how safe it was for a young person. Should we always wear gloves when working with hormones and wash our hands and gloves afterwards? I wasn't clear what precautions are necessary.
Really, really good question. I would take precautions with children. I wouldn't work with liquid auxin because that can absorb through your skin much more easily than the powdered stuff. But then with the powdered stuff, you just want to be careful not to breathe it. But it's really dilute, the powdered stuff. So as long as you're not kicking it up and you have some kind of gloves or you're not touching it, it's much safer, I think, than the liquid form because again, the liquid on your skin can absorb much more easily. So yeah, take precautions, be careful. What I like to do is I like to grow plants, take cuttings at the right time of year when I don't need to use as much hormones. And with kids, you can find plants that root more easily, so you don't even have to add any hormone to them.
All right, Theo F has another question. What do you think about giving fresh cuts to cuttings taken in the field around 2 to 6 days after they were initially taken? Is there a trade-off?
Yeah, so that's a good question because when you make cuttings, you want to apply hormone to the fresh cut. So if you take cuttings in the field and you're keeping them from drying out, then yeah, new fresh cuts where you add hormones to them would help, unless it's something like a grape or whatever, a grape that, those you don't even need to add hormones to them and you could, have them, they'll still root. So it depends, but in general, new fresh cuts are really important. Yeah, because and to clarify, whenever I make cuttings, I cut, put the hormone on there right away. Don't let those cuttings dry out half an hour, 2 hours later when the wounds start healing up, start drying out, and they don't absorb the hormone as much. So yeah, fresh cuts are probably really good. I know of a propagator, Martin Grantham. I had heard that he would carry hormone with him in the field whenever he made cuttings, and he would cut them like in the field and put hormone on there right away. And it's probably for difficult to root things. I would say that's probably the right way to do it.
All right, and we have another question from Portia. Also, how do we time potting up cuttings to ensure it's at the beginning of a root growth period?
Well, in general, roots tend to grow most of the year. They will get stunted when it's really warm. So that's why they're typically better done in the cooler months. But if the top of the plant is actively growing, then that's a good sign that you're going to get root formation. So most plants, except roses, do whatever you want. Okay, grapes probably do, you can do them whatever you want. But most plants, you want to do it when the top of the plant is growing well so that it stimulates the root growth via the auxin production.
All right, we have another question
Uh-huh.
from Kathy C. Do these hormones work the same way or make a difference for taking scions used for grafting versus a cutting going into the ground?
Okay. Well, when you graft things, you don't want to add any hormone because you have the top of the plant, okay, and you have the bottom, right, the rootstock and the scion, and you're taking the auxin from this guy and you're joining it up with the cytokinin from this guy, and it turns out that actually auxin stimulates the cell regeneration that connects the two, okay? So you don't add any hormone when you're grafting. You're letting the two parts communicate with each other.
All right, and this one is from Sienna C. In your list of the functions of ethylene, what are the meanings of adventitious root formation, bromeliad flower induction, and fruit abscission? I will start a botany degree soon, so I'm interested to know what those terms mean.
Okay. You know, I'm gonna have to skip over the ethylene and roots. I haven't looked at that one carefully enough to be able to digest it. Okay, but it turns out that for some reason ethylene at the right amount triggers formation of flowering in bromeliads. There's a compound called ethifon that you put it on there in liquid. It generates this ethylene, and then that stimulates flowering. Why that is, I'm not really I can't digest it. And then there was the third one. I forget what the other one was.
Fruit abscission.
Oh, abscission.
Abscission, yes.
Yes, yeah. So the buildup of ethylene in a plant triggers cell maturation, and then plants will have an area typically at the base of a leaf, but somewhere on the plant that's called an abscission zone, and that's the area where the on one side the cells are going to die, and on the other area side they're not, and so we call that the abscission zone.
All right, the next one is from Franco C. 3 large roots of a mature deodar cypress tree were cut on a construction site. To save the tree, do you suggest any kind of hormone therapy?
That's a really, really tough one. You know, if you can go up to the deodar cedar, if you're worried about drying out, you go in and trim a few of the branches to sort of decrease the water load on it. That could help, but that's a really tough one if major roots were damaged. If there are roots that are under like 2 or 3 inches in diameter, there's a lot other roots that are going to keep feeding the plant. It's when you start getting big trees, when you start getting to the 4 to 6 inch and bigger size, where you can cause some damage to it. So it should be okay if the roots were under like 3 inches in size.
All right, the next 2 are from Mickey R. So we'll go with the first one. Does tomato and blossom plant hormone spray really work to harvest more?
You know what? I don't. I won't speak to the efficacy of it, but if I'd have to see what the compounds in there are to see what it does. Yeah, that's all I could say. You know, typically with tomatoes, if they're over fertilized and they're growing too happily, lots of roots, very vegetative, not as much flowering. So you want to slow the plant down a little bit so it flowers more. And with nutrients, that's why we typically talk about using low nitrogen, more phosphorus and potassium, because nitrogen actually promotes a lot of growth, and that becomes vegetative. So yeah, don't feed the roots too much nitrogen if you want your plant to flower better.
All right, and Mickey's second question. Is there anything I can do to prevent sprouting of buds in potatoes?
I believe it's keeping them in the dark and eating them as fast as possible, or keeping them cool so that they don't sprout. Yeah, the warmer conditions are probably going to lead to more sprouting than cooler conditions.
So in a very cold pantry, maybe. No.
Yeah, or if you could soak them in some kind of if you really have a problem, yeah, cool pantry, exactly. Maybe like soaking them in some kind of compound that was an auxin mimic to prevent cell elongation. Okay, but let's go natural first.
All right, the next one is from Charmaine. Is the meristem in plants analogous to a stem cell in animals?
Oh, yeah, I saw that question earlier and I wanted to touch on that. Yes, stem cells are the same in both organisms. We call them stem cells because they're very foundational cells that can get sent in one direction or the other. And what's cool about plants, I like to joke, is that there's no controversy about the stem cells in plants, okay? Everybody's happy with the meristems. But yeah, there are cells that are undifferentiated. That's why they call them stem cells, like foundational cells. And people are looking at, with respects to taking advantage of those stem cells in humans to help do things. All right. And so I'm not going to get into the ethical questions about that, but they are analogous. Yes.
All right. And we have our last question from Teresa D. How can I top black bamboo and trim the roots so it stops shooting up in our neighbor's yard and grow up under our patio?
How do you feel about Roundup? What you could do is if you've actually bought some kind of auxin compound and you actually treated those stems with lots of auxin, that would inhibit their growth. But it turns out bamboo shoots, when you see a bamboo shoot coming out of the ground, every single cell is already there for the most part, except for if they're going to flower, okay? And so as soon as those shoots come up out of the ground, cut them off. And that will stop that thing from producing for the whole year. So you have to be thorough about it. But if you cut those off when they're young every year, ideally, theoretically, you'll slow it down, okay? But probably getting one of those some kind of auxin compound that you treat the tissue with, you cut them on there. If you don't want to use Roundup to kill them off, you use some kind of auxin compound where you cut them and then treat them with that, and that should sort of really slow them down.
All right. Thank you, Ernesto. Thank you, everyone, for joining us tonight. And we hope you got a lot out of this plant propagation webinar, plant hormones webinar. Again, we're going to have the recording up tomorrow on our YouTube channel. We'll post it on our Facebook and our Instagram. And as well, if you joined our Zoom meeting, you also get an email from Zoom with the information. So just keep an eye out on that. And if you're watching the recording now, hey guys. Ernesto, any final words? Lastly, lots of cytokinin equals shoot growth, and that's why suckers on a plant are not good, because they're going to be very vegetative and not flower for you, so you need to slow them down. But anyways, thank you very much for having me. It's a lot of fun to try to be energetic and try to understand what people are thinking out there while I'm talking. All right. Yeah, you were fantastic. Thank you. And I know we're going to have a lot of views on this later too. Everyone, just again, a last note reminder that next month our meeting will be on Thursday, September 24th, and it will be our Horticulturist of the Year award ceremony here on Zoom at 7:30 p.m. So again, keep an eye out for any information from us in your emails and your social media accounts as well. And thank you again, Ernesto. Thank you to everyone who joined us tonight. I hope you guys have a great night.
Thank you very much for having me.
Bye, guys. You got it.
Bye-bye.